Okay, good morning all. Good morning Shankar, good morning Kusma, good morning Raju, good morning Meda. In group I sent some bits. Okay, try to answer those. Good morning Karthik. Okay, open your group, you find some bits. Okay, open your group, Lakshya Academy WhatsApp group. Okay, you find some bits, try to answer them. Okay, all of you once see your group. Coelozoic parasite lives in. The first bit is coelozoic parasite lives in. Good morning, Vamsi. I am fine. Charita Sri, good morning. Okay, Tirupatamma, good morning. Answer body cavity. Cavities anywhere in the body of the host. First one. Coelozoic parasite lives in. The answer is first one. Good morning, Nageswaram. What is the answer to the second one? Second one. Yes, I had. Cytozoic parasite which lives in the liver cells of man is. Okay, very good, Karthik. The answer is Plasmodium. The second bit answer is Plasmodium. I have sent. Okay, I have sent one photo containing nearly 8 bits in your group. Open that your group and you will find the bits there and try to answer them. The second bit answer is Plasmodium. Next, Nosema notabilis, which is in association with Spirospora, a parasite in the urinary bladder of toadfish is. Third bit. Nosema notabilis is in association with Spirospora, is a parasite in which urinary bladder of toadfish is. Good morning, Pushpalata. The answer is for the third one. Answer quick responses. Okay, very good. Pushpalata gave right answer. Hyperparasite. Good morning, Buddhesh. Next one, enteric rhizopod in man is. Enteric rhizopod in man is. Good morning. Maulali, good morning. Okay, fourth one. Answer fourth one. Okay, third one, hyperparasite. Okay, Meda, right. Okay, very good. Very good. Tirupatamma, Entamoeba histolytica. Fourth answer, enteric rhizopod. Enteric means which lives in the gut. Rhizopods, they have pseudopodia. Okay, we call it lobopodium. The answer for fourth one is, okay, next one. What the answer the fifth one? Digenetic parasite. Okay, leave the word trematode. Trematode you will come across in Platyhelminth, Terbillaria, Trematoda, Cestoda. Digenetic parasite completes the life cycle in man. Answer the fifth one. Okay, Buddhish. Good morning, Venkat Reddy. Fifth one. I am asking fifth one. Digenetic trematode parasite completes the life cycle in two hosts of different species. Answer is digenetic means which completes life cycle in two hosts. Next one, sixth one. Mosquitoes are biological vectors for. Okay, liver fluke. Next one, sixth one. Okay, third one. For fifth one, answer three. Okay, right. Very good, Karthik. Liver fluke. Yes, it completes life cycle in different hosts. It belongs to Trematoda. Mosquitoes are biological vectors for malarial parasite and filarial worm. Malarial parasite and filarial worm. Seventh one, rapid increase in the number of cells due to the effect of a parasite in the host is called. Sixth one is answer one. Seventh one. Rapid increase in the number of cells due to the effect of parasite in the host is. Seventh one, Nageswar Rao. Hypertrophy is not. Hypertrophy increase in the size or volume of the cell. Hypertrophy is increase in the size. Okay, answer is hyperplasia. Very good, Medha Priya and Mohan Sai Reddy. Very good, Janu. Good morning. Okay, next one. Plasmodium causes hypertrophy in. Eighth one. Plasmodium causes hypertrophy too. Okay, right. Medha hyperplasia. Seventh one. Pushpalata, right. Gautam Seshank. Plasmodium causes hypertrophy too. Plasmodium causes hypertrophy to eighth one. Answer is erythrocytes. RBC, correct answer. Answer is erythrocytes in man. Eighth one. Okay, that's all. Evaluate yourself. How many bits you are able to. Okay, you got the correct one. ఓకే ఎక్కడ మీ ప్రాబ్లం ఉందో ఐడెంటిఫై చేసుకోండి ఆ బిట్స్లో ఏ చేయలేకపోయారు అది మళ్ళీ ఒకసారి వాటిని ఐడెంటిఫై చేయండి ఓకే let us start today's class. Okay, we are going to complete the life cycle of plasmodium in human beings. Okay, we are in halfway mark. Yesterday we learned pre-erythrocytic cycle and exoerythrocytic cycle. Today in this class you are going to learn 9 2 marks questions, 9 2 marks questions, 1 4 marks question, that is erythrocytic cycle, and remaining part of the essay. Yesterday you started learning okay one essay, okay, that is erythrocytic cycle, plasmodium life cycle in man, or we can say asexual cycle in man or human phase. Okay, you are in halfway mark. Okay, the remaining part is completed in today's class. Okay, at the end of this period, okay, you will be able to answer 9 2 marks questions, 1 4 marks, and remaining part of the essay as well as MCQs. How can we expect different types of bits from this topic? At the end of this topic, okay, there will be okay bit session also. I will be asking some bits and you will be answering them. Okay, right. Let's get started our today's session. Okay, today's our first topic is erythrocytic cycle or Golgi cycle. Okay, that is our focus. Today's focus is on erythrocytic cycle. Erythrocytic cycle, also called Golgi cycle, also called erythrocytic zygogony. I am using three words here. Okay, I am using three words here. One is erythrocytic cycle, also called Golgi cycle, because Camillo Golgi, okay, described this cycle. The other one is called erythrocytic zygogony. Yesterday too, I used the term as erythrocytic zygogony, which occurs in RBC. Okay, right. Okay, this cycle is completed in the first point I am telling. Okay, this cycle is completed in a span of 48 hours. That means 2 days. Come to the next point here. Next point. Okay, this is the I am starting erythrocytic cycle. Okay, this is a red blood cell RBC. I think you are able to see. It is visible to you. Okay, this is a red blood cell RBC, red blood cell. Okay, now you can observe here, three structures are entering into RBC. Any one of these three can initiate this cycle. Any one of these three. Okay, what are those three? One. Okay, what is this one is? Okay, this is micro meta cryptozoites. Micro meta cryptozoites. Okay, micro meta cryptozoites. Okay, micro meta cryptozoites are formed in which cycle? Exoerythrocytic cycle. At the end of the exoerythrocytic cycle will end, there are macro metacryptozoites and micro metacryptozoites. Here the micro metacryptozoites are may enter into the RBC and initiate the erythrocytic cycle in start stage. The other one, cryptozoite. Okay, where is this cryptozoite is formed? Yesterday we learned. Cryptozoites are formed at the end of pre-erythrocytic cycle. So at the end of the pre-erythrocytic cycle, what are formed? Cryptozoites. Cryptozoites are also called first generation merozoite. They may also initiate the erythrocytic cycle. The third option, at the end of this erythrocytic cycle, okay, there are what are formed here? Merozoites. These are called erythrocytic merozoites. They may also initiate this one. Okay, mainly these two. Another one also there hypnozoites and తర్వాత చెప్తాను. Hypnozoites. Okay, cryptozoites, micrometacryptozoites are formed in exoerythrocytic cycle, and these are formed in pre-erythrocytic cycle. Okay, these two are starting erythrocytic cycle or Golgi cycle. Here the first point I am asking, prati point bit laga adugutunnan. First point, who described erythrocytic cycle? The answer is Golgi. Okay, that hence it is called Golgi cycle also. It is called Golgi cycle. The next one is, okay, how many hours, okay, does it take? It takes 48 hours to complete. How many days? Okay, it is 2 days, 48 hours. Which stages initiate okay, erythrocytic cycle? Erythrocytic cycle is initiated by either cryptozoites of pre-erythrocytic cycle. Okay, Golgi, Camillo Golgi, okay, right? 48 hours or 2 days. And exoerythrocytic cycle of micrometacryptozoites of exoerythrocytic cycle, they may enter any one of these two. They may enter any one of these. Okay, either this one or this one may enter into RBC and start erythrocytic cycle. Okay, next point. This when it enters into the RBC, any one of these three, okay, they attain spherical shape. Now they become in a round shape, spherical shape. Okay, PEC, okay, pre-erythrocytic cycle. EEC, exoerythrocytic cycle. Okay, next, what is formed here? Trophozoite is formed. Okay, up to now you learned 4 times trophozoite stage in Entamoeba histolytica feeding stage. In pre-erythrocytic cycle, it is a feeding stage. In exoerythrocytic cycle, it is a feeding stage. And here also, it is a feeding stage. Feeding stage. Okay, now what it feeds on? It feeds on. Okay, what is this cell? What is this cell? Red blood cell. Inside the red blood cell, there is a respiratory pigment. Can you type in your answer? Can you type in your answer? What is the respiratory pigment present in our RBC? Name the respiratory pigment present in our RBC. Name the respiratory pigment present in our RBC. Okay, very good. Vamsi, hemoglobin is the respiratory pigment. Okay, present in our RBC. Now this trophozoite starts eating. It starts feeding on. It starts feeding on HB. Okay, right. HB hemoglobin. Okay, now it starts feeding on and this one. To feed on hemoglobin, it develops one vacuole called food vacuole. It develops food vacuole. Okay, to feed on our hemoglobin, right? Okay, this vacuole in the initial state, this vacuole is small in size. For example, this is the red blood cell and this is a trophozoite. In it, a small vacuole develops first. Later, this vacuole increases in size. Gradually, it increases in size. When the this what is this cell? What is this one? This is the RBC, red blood cell. What is this one? This is a trophozoite. Okay, this is a trophozoite. What is this one? This is a nucleus. This is the nucleus of the trophozoite. Now, when vacuole develops, now it's clear, I think. Okay, okay. Okay, now here the vacuole develop. This is what is this one? Red blood cell. Inside the red blood cell, what is there? There is a trophozoite. This is the nucleus of the trophozoite. As the vacuole develops in the trophozoite state, the vacuole gradually increases inside. As a result, cytoplasm, this dotted part, this is the cytoplasm, as well as the nucleus are pushed periphery. They are pushed aside. Okay, now it appears like a ring. Okay, because of vacuole, this stage appears like a finger ring. Now this stage is called signet ring stage. Signet ring stage. Okay, this vacuole is nothing but in that vacuole we can put our finger. Okay, that, and here this nucleus is on the side. This is the nucleus of trophozoite. RBC does not has nucleus. RBC, okay, RBC. Which blood cells do not have nucleus all the time? Can you type in your answer? Which blood cells of WBC have nucleus? Question please. Okay, right. Which blood cells? We have different types of blood cells in our blood. Which blood cells do not have nucleus at all? Okay, RBC, mature RBC does not have nucleus, but in immature RBC has nucleus. What is that? Blood platelets. WBC has nucleus. Okay, there is a bilobe nucleus, there is a kidney-shaped nucleus, there is a spherical nucleus, there is a multi-lobe nucleus. There are different shapes of nucleus are present in the WBC. Platelets do not have nucleus. Similarly, mature RBC also does not have nucleus. Here, this nucleus is on peripheral side, on the brim. Okay, this is called, okay, this is the nucleus of not RBC, this is the nucleus of trophozoite stage, which is pushed aside because of a food vacuole. Why it developed? Because it feeds on food. It is for the feeding purpose. What is it feeding on? It is feeding on hemoglobin of. Okay, when it feeds on hemoglobin consists of two components. Hemoglobin has two components. One is Hemoglobin has two components. The one is called heme. One is heme. When there are two components in hemoglobin, I am writing here HB hemoglobin. HB hemoglobin. This hemoglobin has two components. One is called heme. One is heme. The other one is called globin. Hemoglobin is a conjugated protein. Conjugated protein అంటే మీ తర్వాత ప్రోటీన్స్ టైప్స్ బాటనీలో నేర్చుకుంటారు. బయోమాలిక్యూల్స్ అనే టాపిక్ లో వస్తుంది. Hemoglobin is formed by the fusion of two substances. For example, this marker is one hemoglobin molecule. It is hemoglobin molecule. In 10th class, you learned comparison between chlorophyll pigment and hemoglobin pigment. Hemoglobin is a respiratory pigment. Chlorophyll is a photosynthetic pigment. You learned difference. You studied made a comparative study between chlorophyll, photosynthetic pigment, and respiratory pigment hemoglobin. Imagine this is a hemoglobin molecule. Okay, this has two components. One component is called heme. The other component consists of iron. The other component is called globin. Globin is a protein molecule. Okay, this is a protein molecule and this is the heme. This heme consists of iron. Iron is important for the formation of hemoglobin. Now what happens is, now this parasite feeding on our hemoglobin molecules present in the cytoplasm of RBC. But it can digest one thing. It can digest this part, but it cannot digest this part. Which part it can digest? Globin part it can digest. Okay, this is a trophozoite stage, feeding stage. Okay, then it becomes one food vacuole develop. It is eating that. Okay, then it is feeding on that. And then here what is happening is the globin is digested, but on the other hand, heme is not digested. Okay, heme is not digested. This heme appears, okay, next this stage, signature stage, later develops into, look at this one, amoeboid stage. Okay, now I told you 4 stages. For example, if you start this stage, okay, this is మనం ఏమనుకోవచ్చు, మైక్రోమెటాక్రిప్టోజైట్ అనుకుందాం. మైక్రోమెటాక్రిప్టోజైట్ తర్వాత then trophozoite stage తర్వాత signet ring stage. It appears like a finger ring. That's why it is called a ring లాగా ఎందుకు కనపడు, because of development of vacuole. Next one, vacuole disappears. Next, pseudopodial form. Vacuole disappears, vacuole పోతుంది, pseudopodia develop. Now it is called amoeboid stage. It looks like a amoeba. Now it is called amoeboid stage. Okay, it is a signet ring stage. Now it looks like a amoeba. It is called amoeboid stage. This amoeboid stage, what happened? It actively feeds on our hemoglobin. When it feeds on, for example, every day we are taking different types of food. Anni manam tinnayani digest kav. Some of the plant food we cannot digest. Can you name one carbohydrate that we cannot digest? but we are taking every day, but we cannot digest. We does not have any nutritive value. It does not give any energy, but it is very important. Okay, in the absence of that one, we get constipation. Can you tell me one polysaccharide that we take every day, but we cannot digest? Can you tell me one polysaccharide fats are polysaccharide? Polysaccharide that should not digest, but it is important. Presence of it, presence of that food substance in our food is important. That is cellulose. Cellulose. Okay, Raphaeus also we cannot digest. Cellulose. Cellulose is a polysaccharide. We cannot digest because we do not have cellulase enzyme in our body. Cellulase enzyme is not secreted. Okay, right? Here also this this amoeboid stage of the plasmodium, it actively feeds on our hemoglobin. It can digest the globin part. Globin is the protein part. Globin is a protein. Globin is a protein, and it cannot digest the heme. This heme, the undigested heme, is converted to hemojoin. Hemojoin. Hemojoin is called malarial pigment. It is called malarial pigment. It is called malarial pigment. I repeat again. Okay, this is very important part. Hemoglobin has two parts. It is a conjugated protein. Because of combination of two components it is formed. One is called heme, the other one is called globin. Globin is the protein part. Globin is the protein part. It is digested by this amoeboid stage of the plasmodium. But what is the another part? Heme. Heme cannot be digested. That heme okay, this heme is converted to one substance called hemojoin. What is that? Hemojoin ga convert avutundi. Okay, this hemojoin is called malarial pigment. It is called malaria because of this hemojoin. దీని వల్లే మనకి మలేరియా ఫీవర్ వచ్చినప్పుడు మనకి జ్వరం 48 అవర్స్ తర్వాత మనకు ఫీవర్ రావడానికి కారణం మెయిన్ గా హీమోజాయిన్. ఈ హీమోజాయిన్ ఎప్పుడైతే మన బ్లడ్ లోకి రిలీజ్ అవుతుందో ఆర్బిసి రప్చర్ అయిపోయి అప్పుడు మనకు ఫీవర్ వస్తుంది. ఓకే, ద రిలీజ్ ఆఫ్ హీమోజాయిన్ ఇంటూ అవర్ బ్లడ్ ఈస్ అసోసియేటెడ్ విత్ ద మలేరియల్ ఫీవర్. Okay, that's why. Okay, here again I repeat, this is the amoeboid stage. Amoeboid stage, it has pseudopodia. It actively feeds on hemoglobin. It can digest one part, that is the globin part. The other part is not digested, that is the heme part. The heme part is converted to insoluble malarial pigment, that is called as hemojoin. Hemojoin is a malarial pigment which is the heme అనేది heme is converted to hemojoin గా కన్వర్ట్ అవుతుంది. Heme is soluble, whereas hemojoin is a malarial pigment, insoluble crystalline substance. Okay, now amoeboid stage తర్వాత ఈ స్టేజ్ లో ఆర్బిసి సైజ్ బాగా పెరుగుతుంది. అది బాగా తిని the amoeboid stage బాగా సైజ్ లో ఇంక్రీజ్ అవుతుంది. Okay, trophozoite stage also it increases in size. Then amoeboid stage it increase, as a result amoeboid stage ఎప్పుడైతే size increase and our RBC size also increases. That leads to hypertrophy. ఇందా మిమ్మల్ని బిట్ అడిగాను, hypertrophy not hyperplasia. ఇందాక కొంతమంది బిట్ తప్పు చేశారు. ఇది hypertrophy means increase in the volume or size of the cell because of parasite. Here what is this amoeboid state? This is a parasite. Plasmodium is in the form of amoeboid state. Okay, it is present in our RBC size increased because of this parasite, and this is called hypertrophy. Due to this is one of the parasitic effects. We learned gigantism, we learned neoplasia, parasitic castration, hyperplasia. We learned these all things earlier in the effects of parasites on the host. This is the one we learned. hypertrophy, very important. 2 marks lo kuda untundi, 4 marks lo kuda difference between hyperplasia and hypertrophy. Okay, now okay, after tarvata next it becomes increases in size, then it becomes a sigjoint. Baga tini size baga perigi, now it becomes as a sigjoint. Sigjoint you learned in yesterday also in pre-erythrocytic cycle as well as in exoerythrocytic cycle. In pre-erythrocytic cycle also sigjoint is formed from the trophozoite. In exoerythrocytic cycle also sigjoint is formed from the trophozoite. From here sigjoint is not formed from the trophozoite. Okay, sigjoint then is formed in the amoeboid stage nunchi sigjoint formed. ఇక్కడ రెండు ఫీడింగ్ స్టేజెస్ ఉన్నాయి. There are two feeding stages here. One is the trophozoite is the first feeding stage. The other feeding stage is the amoeboid stage is the second feeding stage. Okay, I will tell you some points here. Look at this one. Okay, here Erythrocytic cycle is initiated by cryptozoites of micro, okay, pre-erythrocytic cycle, or micrometacryptozoites of exoerythrocytic cycle. Next one, merozoites from erythrocytic cycle. I showed you those three here. You got merozoites of erythrocytic cycle, cryptozoites and micrometacryptozoites. ఈ ముగ్గురు ఈ Golgi cycle ని initiate చేయొచ్చు. Okay, next one, okay, trophozoite. I am talking about this trophozoite now. Trophozoite is small, round in shape, small, round, and feeding on hemoglobin. It feeds on hemoglobin. Next, early feeding stage. This is the trophozoite is the early feeding stage. What is the another feeding stage? Amoeboid stage is another feeding stage. Next one is the signet ring stage. Signet-ring stage. The important points related to signet-ring stage. Here, vacuole depends in parasite. Okay, vacuole develops. Okay, vacuole develops in the parasite. Eccentric nucleus. Nucleus is in the periphery. That is called eccentric. It is not given in our textbook. Eccentric nucleus అంటే న్యూక్లియస్ అంచులమ్మటి ఉంటుంది, పెరిఫెరీలో ఉంటుంది. నెక్స్ట్ వన్, appears like a ring. That's why it is called signet ring state. It appears like a ring. It is called signet ring state. Those are the features related to signet ring state. I repeat again, vacuole develops here. Food vacuole develops here to feed on RBC, hemoglobin, and the nucleus is eccentric nucleus. That means it is in the side of the cell, not in the middle. Generally, nucleus we find in the center of the cell, but it is in the periphery. Next, it appears like a ring. That's why it is called. It is named as signet ring state. Next, come to the amoeboid state. The points related to amoeboid state. Here, vacuole disappears. Vacuole లేదు ఇక్కడ చూడండి diagram లో amoeboid stage ఇది. This is amoeboid stage. No vacuole here. Vacuole disappears. Next one, centric nucleus. Nucleus is in the center. And next one, formation of pseudopodia. Pseudopodia are formed here. Next one, appears like amoeba. It appears like amoeba. Next one, motile and feeding form. This motile ga untundi. It is also a feeding form. It can move. Motile means it can move. Next one, it feeds on globin. I told you that point. It can feed on globin. It feeds on globin. Hemoglobin lo globin tintundi. It feeds on globin. Next one, protein part. Okay, what is the globin? Globin is the protein part of the hemoglobin. It is the protein part. Globin is the protein part of hemoglobin. Okay, here heme is not digested. The heme is converted to insoluble hemojoin. I told you that one here. Hemojoin. Hemojoin is a malarial pigment. Insoluble. The heme is converted to hemojoin, which is called malarial pigment. Okay, those are the points related to trophozoite stage 3 points, signet ring stage 3 points, Then amoeboid stage, nearly 8 points I mentioned here. Okay, note down those all points. Next, come to the zygont. Okay, now in this zygont, what happens? Yesterday what happened? We learned karyokinesis, karyokinesis as well as cytokinesis takes place. Okay, observe here. Here only one nucleus is present. One nucleus is present. Sorry. Okay, here only one nucleus is present. Okay, now the nucleus divide and form many nuclei, many daughter nuclei. That means karyokinesis occurred here. Okay, karyokinesis occurred here. Okay, this is the nucleus. Here there are many daughter nuclei. Here only one nucleus is present. Okay, then you can observe here some dots I am putting here. Okay, here very important 2 marks question. This one also. This is also 2 marks question. What are these dots I put here? In the cytoplasm of RBC. In the cytoplasm of RBC, there are some dots. These are called Softness dots or Softness granules, which are red colored one. The red colored granules are the red colored dots that appear in the cytoplasm of RBC are called Softness dots or Softness granules. What is the significance of these granules? Scientists look, okay, what they believe is, I told you earlier, two smart parasites. Okay, tell me what are the type in your answer. What are the smart parasites? 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 HIV, very good, Pushpa. HIV as well as Plasmodium is a smart parasite. That means they keep changing surface. Okay, yes, Plasmodium and HIV, Vamsi, very good. Okay, Plasmodium as well as HIV are called smart parasite. Okay, అది ఇప్పుడు ఎందుకు అడిగానంటే రీజన్ ఏంటంటే మీకు అక్కడ చెప్పిన పాయింట్ కి ఇక్కడ రిలేషన్ ఉంది. Okay, ఇది what is this one? Plasmodium in the form of amoeba. Okay, this Plasmodium releases some antigen. Those antigens are released into our cytoplasm of RBC. Okay, again it forms a new antigen. Okay, the scientists believe that these Softness dots or Softness granules are the antigens produced by the amoeboid stage into our Plasmodium into the cytoplasm of our RBC. It is 2 marks. Yes, HIV. Okay, Pavan Teja. Okay, right. HIV and Plasmodium. Okay, what are Softness dots or Softness granules? What is their color? They are red in color. Where are they present? They are present in the not cytoplasm of this amoeboid. They are present in the cytoplasm of the red blood cell. Okay, what scientists believe that these are the antigens produced by the parasite. Okay, the parasite produce new antigen. Okay, those new antigens are they are released into the cytoplasm. Okay, that's why it is called smart parasite. Okay, observe here, this is the nucleus. And what are these dots here? These are called hemojoin granules or hemojoin pigment. Hemojoin pigment. ఎందుకు ఇక్కడ ఉందంటది తను డైజెస్ట్ చేసుకున్నది గ్లోబిన్, తను డైజెస్ట్ చేసుకోలేని పార్ట్. ఫర్ ఎగ్జాంపుల్ ఎవ్రీ డే we are eating food, some of the food is coming outside in undigested form. Like that, this amoeboid stage also, it can digest some part of the hemoglobin, that is globin. It cannot digest the remaining part, that is globin part. It remains as the name we call hemojoin. Now come to the zygont stage. ఇది బాగా తినేసి size increase అవుతుంది. Okay, this is called zygont stage అంటాం. Now this zygont stage ఏమవుతుంది, ఇక్కడ karyokinesis and cytokinesis takes place. Okay, now very important point coming here. Okay, this is observe these structures here. Observe these structures here. Now it is looking like a rose flower, petals of rose flower. This stage is called Rosetti stage. Rosetti stage. Because what are these? These all are nuclei. What is the cytoplasm? Cytoplasm got divided. We turn it merozoites. These are called merozoites. The merozoites, how many merozoites are formed? 12 to 24 merozoites are formed. These merozoites arrange in the form of rose petals of a rose flower. That's why this stage is called Rosetti stage, amoeboid stage, signet ring stage, amoeboid stage. This is the Sison stage, and this is the Rosetti stage. These are the new terms. This as it appears like ivanni kuda merozoites any from 12 to 24. Those merozoites inside the RBC, they are arranged in the form of petals of a rose flower. That's why it is called Rosetti stage. Rosetti stage. Now finally what happens is the RBC ruptures. The RBC rupture and releases. Two things are released from it. Very, very important. Listen carefully. This is the red blood cell. The red blood cell ruptured, and finally what are released here are erythrocytic merozoites. You learned first generation merozoite, second generation merozoite. Merozoites at the end of the first the pre-erythrocytic cycle, and merozoites at the end of the exoerythrocytic cycle. Now here also merozoites are formed. These are the third generation coach one. These are called erythrocytic merozoites because these are formed in erythrocytic cycle. Erythrocytic merozoite. Now along with that, what is released? This is called hemojoin. Is also, this is a malarial pigment. When these RBC, not one, just we studied one RBC, like that many RBC are ruptured, and this malarial pigment is released. ఎప్పుడైతే malarial pigment మన blood లోకి రిలీజ్ అవుతున్న RBC rupture అయ్యి, at that time that person gets fever. I told you in the beginning here, this cycle is completed in 48 hours. In the beginning of this topic, Plasmodium, I told you, malaria may come after 48 hours or 72 hours అని చెప్పుకున్నాం. 48 hours తర్వాత అంటే థర్డ్ డే వస్తే దాన్ని ఏమనుకున్నాం, టెరిస్టియన్ ఫీవర్. అదే ఫోర్త్ డే వస్తే క్వార్టన్ అనుకున్నాం. మరి ఇప్పుడు మనం చదివేది అంతా కూడా దిస్ ఈజ్ అబౌట్ ప్లాస్మోడియం వైవాక్స్. ఇన్ ప్లాస్మోడియం వైవాక్స్, this erythrocytic cycle is completed in 48 hours 2 days. That is the reason why in that person who has this Plasmodium vivax in his body, he gets fever on the third day. After a gap of 48 hours. What is happening in this 48 hours? Erythrocytic cycles take place, and this at the end of those 48 hours, hemojoin is relieved. That means he is getting fever on the third day. Next, one more important point I told you earlier. Here anemia also. Malarial patient also develops anemia. Why? Because anemia means less number of RBC. Here the Plasmodium is attacking our RBC, or RBC are destroyed. Our RBC are destroyed. That's why here the number of RBC also falls. The number of RBC also falls in the malarial patient. అందుకే మలేరియల్ పేషెంట్ కి మలేరియా బాగా సివియర్ ఇఫ్ ఇట్ రిమైన్స్ అన్ట్రీటెడ్ ఫర్ ఏ లాంగ్ టైమ్, హీ రిక్వైర్స్ ఫ్రీక్వెంట్ బ్లడ్ ట్రాన్స్ఫ్యూజన్స్. అతనికి రక్తాన్ని ఎక్కించాలి, బికాస్ అతని బాడీలో ఆర్బీసీ నెంబర్ బాగా గ్రేట్ గా ఫాల్ అయిపోయింది. ఆ ఫాల్ కావడానికి కారణం ఏంటంటే బికాస్ ఆఫ్ రిపీటెడ్ ఎరిథ్రోసైటిక్ సైకిల్. Like that, these merozoites again enter into the RBC, or after repeated cycles, nearly 8 to 9 cycles, a number of cycles jarigin tarvata, very very important here, some of the merozoites enter into RBC and they become female gametocytes, and some of the merozoites enter into some other RBC, and they form male gametocyte. This conversion of merozoites into gametocytes take place in the spleen, when the RBC are in the spleen and bone marrow. spleen and bone marrow. Okay, I repeat the point మళ్ళీ చెప్తున్నాను. Okay, these are called merozoites. ఈ black color లో డ్రా చేసిన these are called merozoites. Merozoites okay ఏమవుతాయంటే okay they enter into RBC and they start another erythrocytic cycle. ఇలా చాలా cycles జరిగిన తర్వాత after number of erythrocytic cycles okay మన బాడీలో RBC నెంబర్ బాగా గ్రేట్ గా డిక్రీజ్ అవుతుంది. Okay, at that time some of the merozoites enter into RBC and they transform into okay some of them transform into female gametocytes. Female gametocyte is larger in size and some of the gametocytes enter into RBC and they transform into male gametocyte which is smaller in size. Okay, this is the larger in size female gametocyte. The male gametocyte is smaller in size. Okay, these gametocytes cannot continue in our body further because okay, in our body they cannot develop in further stages ga develop kalevu endukante our body temperature and our pH value are not suitable for its further development. That means now they are ready to enter into the another host. Okay, ee manlo cycle ayipindi man ante we are called secondary host or intermediate host. Ippudu now it is waiting for the another host. Okay, when these RBC okay during the night time these RBC with gametocytes enter into our peripheral blood circulation. Peripheral circulation means mana skin ki kinda una circulation loki vastayi night time. Okay, when mosquito bites that malarial patient during night time okay, they enter into the mosquito lopaliki enter. They enter into the crop of the mosquito. When it feeds, when it sucks our blood, it enter into the stomach or crop of the mosquito lopaliki enter. Okay, mari doma lopaliki only gametocytes e kaadu, amoeboid stages may also enter into the mosquito and okay, sison stage also may enter, signet ring stage also may enter, trophozoite stages also may enter into the mosquito. Kani these all are digested. These all stages, for example doma, for example I have malaria. If I have malaria during night time into my peripheral blood circulation ante skin ki daggaraga unna blood circulation loki ఈ female gametocyte male gametocytes ఉన్న RBC they come into my peripheral blood circulation. Sir, why tree there is no effect on mosquito too is affected. Mosquito lo stomach wall crop wall damage అవుతుంది, అందుకే దాన్ని మనం ఏ మస్కిటో అంటాం, ఇన్ఫెక్టెడ్ ఫీమేల్ ఎనాఫిలిస్ మస్కిటో. Not all mosquitoes. For example, if I am bitten by a normal female anopheles mosquito, infected can't, I don't get malaria. Only infected female anopheles mosquito ma pudite matrame manaku vastundi. Aa mosquito kuda damage untundi, mosquito యొక్క crop wall damage avutundi. Ronald Ross on the first day I told you, Ronald Ross observed okay oocysts of the Plasmodium on the crop wall. దాని క్రాప్ వాల్ కూడా డ్యామేజ్ అవుతుంది, మస్కిటో కూడా కొంత డ్యామేజ్ అవుతుంది, బట్ ఇట్ డస్ నాట్ గెట్ మలేరియా. Okay, right. These male gametocytes and female gametocytes which are present in RBC, when mosquito sucks blood, okay, they enter into the primary host or invertebrate host or definitive host, and there it completes the sexual cycle. Okay, ఎప్పుడైతే దోమ బాడీలోకి మన RBC ఎంటర్ అవుతాయో RBC డైజెస్ట్ అయిపోతుంది. RBC డైజెస్ట్ అయిపోయినప్పుడు ఆ RBC లో నుంచి ఎవరు బయటకు వస్తారు? In the crop of the mosquito, our RBC are digested by enzyme, and what come outside? Male gametocyte and female gametocyte. Okay, the remaining development we study in the next one. Okay, this is about erythrocytic cycle. Okay, what ఇవాళ మనం చెప్పుకున్న లెసన్ లో నిన్న చెప్పుకున్న లెసన్ లో వచ్చిన కొన్ని 2 marks క్వశ్చన్స్, why not is that morning time? Why not? Because it is suitable for okay, for example if you take in case of filariasis కి సంబంధించి క్యూలెక్స్ మస్కిటో అని చెప్పుకున్నాం ఇంత ముందు. ఓకే, ఆ దోమలు నైట్ night 10 to 14 మార్నింగ్ 4 నైట్ 10 టు మార్నింగ్ 4 టైంలో ఆ ఒకర్ ఏరియాకి సంబంధించిన స్టేజెస్ ఆ పేషెంట్ బాడీ బ్లడ్ సర్క్యులేషన్ లోకి వస్తుంది. క్యూలెక్స్ మస్కిటో ఈ బైట్స్ నైట్ బిట్వీన్ నైట్ 4 నుంచి 10 మార్నింగ్ 4 వరకు. దట్ ఈస్ కాల్డ్ నాక్టర్నల్ పీరియాడిసిటీ. తర్వాత వస్తుంది మీకు అది నాక్టర్నల్ పీరియాడిసిటీ. Okay, why? Because night time that is suitable to enter into the, okay, because female Anopheles mosquito does not bite us during the daytime. Okay, Aedes mosquitoes mana pagalupotu kodutayi. Okay, female Anopheles mosquito bites during night time. If these gametocytes come to the peripheral circulation during the daytime, they have to enter into the mosquito within 1 week. These gametocytes, they have to enter into the body of the mosquito within 1 week. That means 7 days. Otherwise, ఆ gametocytes మన బాడీలోనే చనిపోతాయి. అంటే అది నెక్స్ట్ హాస్ట్ లోకి అవి ట్రాన్స్ఫర్ కావు. ఓకే, హియర్ ఆర్ ద 2 మార్క్స్ క్వశ్చన్స్ వాట్ వీ హ్యావ్ లెర్న్. నిన్న క్లాస్ లో కానీ ఇవాళ క్లాస్ లో కానీ నేర్చుకున్నాయి నేను చెప్తున్నాను. వాటి మీనింగ్స్ ఏంటి, మీకు ఇంకా కొన్ని తెలియని ఏమైనా ఉన్నాయో చూడండి. ఫస్ట్ వన్, ఓకే, మనకి 23rd క్వశ్చన్ నుంచి మనకి 2 మార్క్స్ స్టార్ట్ అవుతాయి. ఐ విల్ టెల్ యూ దోస్, ఐ విల్ సెండ్ యూ దీస్ 2 మార్క్స్ క్వశ్చన్స్. 9 అని చెప్పాను, ఇవాళ క్లాస్ లో మనకి దాదాపు 9 కంప్లీట్ అవుతాయి. ఫస్ట్ వన్, వాట్ ఆర్ ద స్టేజెస్ ఆఫ్ ప్లాస్మోడియం వైవాక్స్ దట్ ఇన్ఫెక్ట్ ద హెపాటోసైట్స్ ఆఫ్ మ్యాన్? Very important. What are the stages of Plasmodium vivax that infect the hepatocytes of man? That means liver cells of man ni infect chese Plasmodium vivax stages enti. Can you type in your answer? Me answer ni type chesi pampagalra quick ga. What are the stages of Plasmodium vivax? మీ దగ్గర ఉన్న మెటీరియల్ ఏమైనా ఉంటే చూసి ఆన్సర్ చేయకుండా, what are the stages of Plasmodium vivax that can infect the hepatocytes of man? Right, very good. Sporozoite, Vamsi, 1 is correct. Sporozoite is 1. Inka some more are there. Okay, Sporozoite, Syed, Buddhesh, Nageshwara, Vamsi, Sporozoite is correct. What are the other stages that can infect liver cells? How to write class notes? Janu Reddy asking how to write class notes. At the end of the class, you replay this video and you can write these all points. You can note down these all points, and I will send you other notes also. Signet ring stage. What are the stages that can infect liver cells or hepatocytes? Sporozoite, 1 correct answer has come. 2 more are there. Okay, I am telling here. Amoeboid stage and Anopheles mosquito, no. One is Sporozoite. Another one is cryptozoites at the end of the pre-erythrocytic cycle. For example, this is the, I am drawing rough diagrams here. This is a sporozoite, sickle shaped sporozoite entered into it. Then it transforms into trophozoite. Then it transforms into cyjon, larger one. And then it undergoes cyzogony. It undergoes cyzogony. And these are all the daughter cell. And it, the red blood cell as well as the cyzon burst and releases. And what they release? Okay, these, what do we call these stages? Cryptozoites. These cryptozoites also, yesterday I told you, cryptozoites again enter into, they have 2 options. One is to enter into the RBC, red blood cell. I told you today also, Okay, they may enter into the RBC. The other one is they may enter into the liver cell. That means cryptozoites are also entering into the liver cell, not only sporozoite. Understand? Cryptozoite. Next one. At the end of this stage, next the cryptozoite entered here. Cryptozoite entered into the liver cell. Then it transforms into trophozoite. Then it transforms into trophozoite transforms into scission. Scission very large in size. Then it undergoes karyokinesis and cytokinesis. At the end of this one, again water form. At the end of this one, water form. Micrometacryptozoites and macrometacryptozoites. The macrometacryptozoites may again attack the fresh liver cell. Okay, the answer is 3 stages here may and attack the liver cells. Okay, micrometacryptozoites. No, micrometacryptozoites attack the micrometacryptozoites. They attack the in the micrometacryptozoites. They may attack the liver cell RBC. Micro they attack the RBC. Macro also attack a tarvata vastundi. Okay, here these macrometacryptozoites again attacking liver cell. Here sporozoite is attacking liver cell, cryptozoite is attacking liver cell, and macrometacryptozoite also attacking liver cell. The answer is this one. Okay, this is the one of first 2 marks question from this topic, Plasmodium. Okay, I repeat again. What are the stages of Plasmodium vivax? Okay, that infect the hepatocytes, sporozoite, cryptozoite, and macrometacryptozoite. I hope it is clear. Now come to the next one. What are the stages of Plasmodium vivax? ఇప్పుడే ఇవాళే చెప్పారు. What are the stages of Plasmodium vivax that infect the RBC of the intermediate host? That means we are the intermediate host or secondary host or vertebrate host. మన ఆర్బీసీని అటాక్ చేసే స్టేజెస్ ఏంటి? Okay, tell me your answers, send your answers. Nagalakshmi. Which stages attack our RBC? Ival cheppindi. What are the stages? Yes, micrometacryptozoites. Very good, Medha Priya. Micrometacryptozoites, that is one. Lohita Reddy, she is telling 2 stages. 3 are there. 3 are there. Observe here. Erythrocytic merozoites. Erythrocytic merozoites are attacking RBC. Next one, I wrote 2 things here. Cryptozoites. Cryptozoites and the first generation merozoites may attack the RBC. Next one, the other one is micrometacryptozoites also may attack the RBC. Sir, doubt Nagalakshmi. What's your doubt? Medha Priya, merozoites, cryptozoite, merozoite. Okay, the answer is cryptozoites are first generation merozoites and micrometacryptozoites of exoerythrocytic cycle. And next one, this one. One more is also there, hypnozoites. Hypnozoites. What are hypnozoites? Hypnology, hypnologist, hypnozoites. Aedes mosquito means AEDES spelling, Aedes mosquito. Aedes mosquito is one type of species. There are different species of mosquitoes. Culex is one species, Anopheles is one species, Aedes, AEDES, Aedes is one species. ఈ దోమలు డెంగీ ఫీవర్, చికెన్ గున్యా ఇలాంటి వైరల్ డిసీజెస్ ని స్ప్రెడ్ చేస్తాయి. జనరల్లీ ఎడిస్ మస్కిటోస్ బైట్ డ్యూరింగ్ ద డే టైం. అండ్ ఎవరైతే చికెన్ గున్యా, డెంగీ ఫీవర్ ఉన్న వాళ్ళని ఈ దోమలు కుట్టినప్పుడు వాళ్ళ బాడీ లోపలికి, ఆ మస్కిటో లోపలికి వైరస్ వెళ్ళి ఇంకోరికి అది వైరస్ స్ప్రెడ్ అవుతుంది. సార్ హెపాటిక్ సైకిల్ ఒకసారి చెప్పండి. హెపాటిక్ సైకిల్. Okay, did you attend yesterday's session or not? I don't know. I am seeing your name first time, Nagalakshmi. If you want hepatic cycle, yesterday's videos you can see, or videos are there in Laksha YouTube channel. Okay, if you want that, okay, if you want me to put that link in our number, if you send your number to me, I will send that yesterday's video. You can again watch it. You can replay it. Okay, the answer is what? Now I am explaining what are hypnozoites. Listen carefully. Hypno means hypnology. Some of the macrometacryptozoites, and they formed in exoerythrocytic cycle. Some of the macrometacryptozoites, they may remain in dormant stage. Okay, you learned in 10th class seed dormancy. Seed dormancy is induced by, can you type in your answer, which phytohormone, which plant growth substance can induce, okay, dormancy in seeds? Which plant hormone induces dormancy in seeds? రెస్ట్ లో ఉంది, తర్వాత యాక్టివేట్ అవుతుంది కదా సార్? ఎస్, వెరీ గుడ్. Okay, abscisic acid, very good, absolutely correct. Abscisic acid is a phytohormone or plant growth substances, a plant growth regulator, which can induce dormancy in the seeds and which can cause the falling of the leaf, shedding of the leaf. Okay, here also, and these macrometacryptozoites of exoerythrocytic cycle, sometime the macrometacryptozoites of the exoerythrocytic cycle, they remain in a dormant stage in our liver. Nidrapotu unte not gibberellin. They remain. Ethylene also one of the phytohormone. Auxin is one of the phytohormone. Ethylene, auxin, gibberellin, cytokinin, abscisic acid, ethylene. Okay, these are the hormones in the plants, phytohormones. Okay, right. Okay, some of the macrometacryptozoites, they enter into the dormant stage and they remain in the inactive, okay, state in the liver. When they activated, all of a sudden, they suddenly activate what? Macrometacryptozoite. And when they are reactivated and they again start the erythrocytic cycle, start just not the exoerythrocytic cycle. They start the erythrocytic cycle. అంటే మొత్తం ఇప్పుడు erythrocytic cycle ని 4 members start చేయొచ్చు. 4 members. Answer what? For what? Answer is for what? Okay, abscisic acid induces dormancy. Here hypnozoites అనేవి ఏంటంటే, what are hypnozoites? It is 2 marks question ఇది. Hypnozoites are the macrometacryptozoites. Macrometacrypto మొత్తం 4 ఉన్నాయి. 4 అటాక్ చేయగలవు RBC. One is cryptozoites are first generation merozoites. Cryptozoite, this one, and second cryptozoite, and third one erythrocytic merozoite. And what is the another one is hypnozoites. Hypnozoites and macrometacryptozoites which remain in dormant state. All of a sudden, that is called relapse of malaria. For example, if నాకు మలేరియా ఉంది, మలేరియా మెడిసిన్ వాడితే తగ్గిపోయి కొన్ని రోజుల తర్వాత నా లివర్ లో కొన్ని మ్యాక్రోమెటాక్రిప్టోజైట్స్ డార్మెంట్ స్టేజ్ లో నిద్రపోతూ ఇన్యాక్టివ్ గా ఉంటాయి. ఇఫ్ అవి మళ్ళీ రీయాక్టివేట్ అయితే again I get malaria. That is called relapse of malaria because of reactivation of macrocryptozoites initiate the fresh erythrocytic cycles, but they enter liver cell now. Okay, next. Okay, general గా జరిగితే లివర్ సెల్ లోకి ఎంటర్ అవుతుంది. ఇలా అవి హిప్నోజైట్స్ గా కన్వర్ట్ అయితే they enter into the RBC. That means macrometacryptozoite generally they enter into the liver cell. In case of adi hypnozoites ga convert aithe, they start the erythrocytic cycles. That is called relapse of malaria. Okay, next one is define pre-patent period and what is its duration in the life cycle of Plasmodium. Okay, time is already 12:37. I am going to explain you two periods, pre-patent period and incubation period. What are the two? These two. Pre-patent period and what is incubation period? Okay, this session may take still 15 minutes, okay, to explain these questions so that you can understand. I will send it to you. Then you can learn and easily and you can write it. Okay, what is incubation period? That is 1-2 marks. What is pre-patent period? What is pre-patent period? First one. Okay, this is the time interval between the first entry of Plasmodium into our blood in the form of దోమ కుట్టినప్పుడు ప్లాస్మోడియం మన బాడీ లోపలికి ఏ ఫామ్ లో ఫస్ట్ ఎంటర్ అవుతుంది ఇన్ ద ఫార్మ్ ఆఫ్ స్పోరోజాయిట్. That is sporozoite. It enters into our body in the form of sporozoite. This is the first entry. Then it goes to liver cells. It goes to liver cell. It stays there nearly 8 days. 8 days ante pre-erythrocytic cycle. And after 8 days of period, after 8 days, okay, first time ఇది మన బ్లడ్ లోకి ఎంటర్ అయింది స్పోరోజైట్ దోమ కుట్టినప్పుడు. After 8 days again it enters into the blood in the form of cryptozoites. ఈ టైం ఈ 8 days ఇది ఎక్కడ ఉందంటే లివర్ సెల్స్ లో ఉంది. Okay, this period is called pre-patent period. Pre-patent period is 8 days. That means okay, pre-erythrocytic cycle duration untundo, that is the pre-patent period. I repeat again, pre-patent period. For example, let us take one hero. Okay, there is one hero in the one movie. A movie starting లో హీరో ఒక అంటే డబుల్ రోల్ ప్లే చేస్తున్నారు. ఓకే, ఒక రోల్ లో ఎంటర్ అయ్యాడు. ఫస్ట్ ఎంట్రీ ఆఫ్ ద హీరో ఇన్ ద ఫార్మ్ ఆఫ్ సమ్ యాక్షన్ ఏదో ఒక ఫామ్ లో. నెక్స్ట్ ఇంటర్వెల్ తర్వాత హీరో ఇంకొక ఫామ్ లో ఎంటర్ అయ్యాడు. అంటే ఫస్ట్ ఎంట్రీ ఇన్ వన్ ఫామ్ అండ్ సెకండ్ ఎంట్రీ ఇన్ అనదర్ ఫామ్, డ్యూయల్ రోల్. ఓకే, ఇలా కూడా ప్లాస్మోడియం ఫస్ట్ మన బ్లడ్ లోకి ఏ ఫామ్ లో ఎంటర్ అయింది దోమ కుట్టినప్పుడు స్పోరోజైట్ ఫామ్. ఆఫ్టర్ 8 days, okay, remain 8 days it remains in liver cell. After 8 days again they release into the sinusoids of the liver. In sinusoids లో బ్లడ్ ఉంది, మళ్ళీ 8 రోజుల తర్వాత మళ్ళీ బ్లడ్ లోకి వస్తుంది. This is the first entry, this is the second entry. ఈ రెండు ఎంట్రీస్ మధ్యలో ఉన్న టైం ఇంటర్వెల్ ని we call pre-patent period. I repeat again, the time interval between the first entry hero first entry. Okay, అంటే ప్లాస్మోడియం, ఇమాజిన్ ప్లాస్మోడియం is one hero. మన బాడీ లోపలికి ఎంటర్ అయ్యాడు. ఏ ఫామ్ లో ఎంటర్ అయ్యాడు? స్పోరోజాయిట్ ఫామ్ లో. within half an hour they reach the liver cell. They stay in the liver cell for 8 days. After 8 days again the hero is entering again into the blood. Okay, the first entry in the form of sporozoite and the second entry in the form of cryptozoite. In between these two entries there is 8 days time. This is called pre-patent period. Time is up, I know. Apparao boy party, time is up, I know. Okay, sometimes we have to extend our okay, there is no time limit for learning. There is no time limit for learning. Learning is a journey. You cannot stop that journey. If you stop once your learning journey, our journey, life journey also ends there. Okay, right from the beginning, right from the childhood, when you are born, from that time onward till we die, our learning goes on. We learn outside the classroom, we learn inside the classroom. Okay, there is no end to learning journey. Okay, more you learn, more you earn knowledge, more you earn, more marks you can score, better mark, okay, rank you can get. Okay, my words may be boring to you, but that is the reality. Okay, he retracted his words, he retracted his message after listening my words. Okay, now come to the incubation period. What is meant by incubation period? Okay, after the end of this incubation period, I am going to stop my session. Don't worry. Okay, what is incubation period? Okay, this is the first entry of sporozoite into our body. Okay, sporozoite entered into our body in the first stage. This is the entry. After, again, the sporozoite is released into our body. Sorry, the other stages are releasing into our body, which is causing malarial fever. What is that? Here, hemojoin is releasing. When hemojoin is released, at that time you get fever. That means the fever first pre-erythrocytic cycle complete. That means 8 days. Next one, exoerythrocytic cycle also maybe. Next one, erythrocytic cycle 48 hours, 2 days. And 8 days plus 2 days. That means 10 days. For example, if a person is bitten by a mosquito on the 1st July, ఫస్ట్ జూలై దోమ కుడితే అతనికి మలేరియా ఏ రోజు రావచ్చు అంటే ఆగస్ట్ 10th నుంచి 14th లోపు జ్వరం రావచ్చు. ఓకే, ఈ పీరియడ్ ని వి కాల్ ఇంక్యుబేషన్. ఎలా మనం చెప్పగలం అంటే అతనికి ఎప్పుడైతే మలేరియల్ సిమ్టమ్స్ అపియర్ అవుతాయో, ఓకే, అప్పుడు అతనికి ఇంక్యుబేషన్ పీరియడ్ కంప్లీటెడ్, మేనిఫెస్టేషన్ పీరియడ్ స్టార్ట్ అయింది. ఐ రిపీట్ అగైన్, దేర్ ఇస్ నో క్లారిటీ హియర్. Incubation period is the time interval between the entry of sporozoite into our body and first appearance of malarial symptom. That is called incubation period. It is 10 days to 14 days. 10 days to 14 days. It may vary. It is 10 days to 14 days. Okay, అంటే మనం we have to include here pre-erythrocytic cycle, exoerythrocytic cycle and erythrocytic cycle. ఇవన్నీ యాడ్ చేస్తే మనకి 12 days వస్తుంది. Around 10 to 12 days, that is the incubation period. These two are the very very important. These two are the 25 and 26 questions in our material. Okay, the next one is what are Softness dots? What are Softness dots? Okay, red colored granules. What are Softness dots? The red colored granules that appear in the cytoplasm of Rbc of man when the Plasmodium parasite remains in the amoeboid state. When the Plasmodium is in amoeboid state, in the cytoplasm there are red colored structures are present. These are called Softness granules, are the dots which are believed to be the antigens released by the parasite. Okay, these dots are believed to be the antigens produced by the Plasmodium to stimulate the host to produce antibodies. Next one, what are hemojoin granules? What are hemojoin granules? This is the 28th question. In the erythrocytic cycle of Plasmodium, the parasite ingests hemoglobin, of which globin part is digested. I told you earlier here, globin part is digested and convert the soluble heme into insoluble crystalline hemojoin. Okay, that is called hemojoin. Hemojoin is a malarial pigment. It is called malarial pigment. When it is released, the person gets fever. Okay, right. Come to the last part of today's class. Last part. It is not given in your textbook. It is very important. Please, I request you to listen this table. Write down this table. It is not given in your textbook. Okay, next, in the beginning of, on the first day of this topic, and that means day before yesterday, I told you 4 species of Plasmodium. Okay, these are the 4 species of Plasmodium: Plasmodium vivax, Plasmodium ovale, Plasmodium malarium, and Plasmodium falciparum. And the type of malaria they cause also I explained you day before yesterday. Okay, now as I completed today incubation period, I have written here incubation periods of these 4 species against these 4 species here. The incubation period of vivax is 14 days. Okay, మనం ఈ లెసన్ లో చదివినంత కూడా vivax, Plasmodium vivax incubation period 14 days. The incubation period of Plasmodium ovale is 14 days. The incubation period of Plasmodium ovale is 28 days. 28 days. Next one, the incubation period of Plasmodium falciparum is 12 days. That means incubation period is not same for all the species of Plasmodium. It varies. Come to the duration of erythrocytic cycle. Okay, erythrocytic cycle. మీకు ఇంతకుముందు క్వార్టర్ మలేరియా, క్వార్టర్ మలేరియా, టెరిషియన్ మలేరియా అని చెప్పాను. అది ఎందుకు ఆ పేరు వచ్చిందో చెప్తున్నాను. ఓకే, డ్యూరేషన్ అన్నారు. ఇక్కడ డ్యూరేషన్ అంటే ఎరిథ్రోసైటిక్ సైకిల్ కి అన్ని స్పీస్ కి ఒకే టైం పట్టదు. ఫర్ ఎగ్జాంపుల్, ఇఫ్ యూ టేక్ ప్లాస్మోడియం వైవాక్స్, the erythrocytic cycle duration is 48 hours. That means 2 days. Next one, for Plasmodium ovale 48 days. ఇది 48 days hours, 48 hours at 2 days తర్వాత ఫీవర్ వస్తుంది కాబట్టి దిస్ ఈజ్ కాల్డ్ టెరిషియన్ మలేరియా. బినైన్ అంటే అంత హార్మ్ఫుల్ కాదు. నెక్స్ట్ వన్, ప్లాస్మోడియం ఓవేల్. ద డ్యూరేషన్ ఆఫ్ ఎరిథ్రోసైటిక్ సైకిల్ డ్యూరేషన్ ఈజ్ 48 hours. దట్ మీన్స్ 2 days. ఇట్ ఈస్ ఆల్సో టెరిషియన్ అని పేరు పెట్టారు ఎందుకంటే థర్డ్ డే ఫీవర్ వస్తుంది. ఆఫ్టర్ 48 hours తర్వాత 3వ రోజు జ్వరం వస్తుంది. It is mild, not anta harmful kaadu. Plasmodium ovale. Next, Plasmodium malariae. 72 hours. 72 hours is the erythrocytic cycle. అందుకే దీని అంటే 72 hours means 3 days. 4వ రోజు జ్వరం వస్తుంది దీనికి. 72 hours కంప్లీట్ అయిపోతున్న తర్వాత 4th డే జ్వరం వస్తుంది కాబట్టి దీన్ని క్వార్టర్ మలేరియా. క్వార్టర్ మీన్స్ 4. క్వార్టర్ అంటే 1/4 అంటే ఇక్కడ 1/4 అంటే 4th డే ఫీవర్ వస్తుంది. దట్స్ వై ఇట్ ఈస్ నేమ్డ్ as క్వార్టర్ మలేరియా. The next one is falciparum, which is dangerous అని చెప్పాను. ఎందుకు ఇది dangerous? It causes the malignant malaria, or cerebral malaria, which is dangerous among the 4. The most common is this one. The most dangerous is this one. Why it is dangerous? The reason is here. 1, its incubation period is compared to 4 species here. It has a lowest incubation period covered. It causes the greatest harm. Come to the duration of the erythrocytic cycle. Its erythrocytic cycle duration is 36 to 48 hours. Look at here, 48 hours, 72 hours. Here 36 to 48. మరి ఇంక్యుబేషన్ పీరియడ్ డ్యూరేషన్ ఆఫ్ ఎరిథ్రోసైటిక్ సైకిల్ డిక్రీజ్ అయితే వాట్ ఈస్ ద మనకి హార్మ్ ఎలా అంటే 36 hours లో మన ఆర్బీసీని డెస్ట్రాయ్ చేయగలదు. బట్ ఇట్ 48 hours లో అంటే డెస్ట్రక్షన్ ఆఫ్ ఆర్బీసీకి హియర్ ఇట్ ఈస్ టేకింగ్ మోర్ టైం. But it is within a span of 36 to 48 hours, it is causing damage to our RBC. That's why it is most dangerous one. And it causes subterranean malaria. Sub అంటే below, అంటే టెరిషన్ అంటే 3 రోజులకు ముందే, అంటే 3వ రోజు రాకముందే జ్వరం రావచ్చు. అంటే 36 hours తర్వాత ఫీవర్ రావచ్చు, లేదా 48 hours. That's why it is called subterranean. ఇది టెక్స్ట్ బుక్ లో ఇవ్వలేదు. Okay, this table, write down this table. It is very important. I hope it is worthy and it is very clear to all of you. Okay, tomorrow's class, remaining part of the plasmodium will be completed in tomorrow's class. Okay, that's from my side today. Okay, that's the end of the session. Okay, bye to all of you. I too thought the same. Okay, very good. Hari Prasad ready. Okay, so many students, they are sending their work. They are working very hard and they are writing. The homework what I allotted to them, they are finishing and sending to me. Once again, I repeat my mobile number. Okay, please send your work. Any questions? And if you pamputanu. Okay, 9 4 marks questions. I am going to send you 9 4 mark questions. 9 4 marks 9 2 marks 1 4 mark 1 essay. ఓకే, ఆ ఎస్ఏలో కొంత పార్ట్ నిన్ను కంప్లీట్ చేశారు. రిమైనింగ్ ఇవాళ వర్క్ పంపుతాను, అది కంప్లీట్ చేయండి. కంప్లీట్ చేసి ఈ నెంబర్కి మీరు అది మెసేజ్ చేయండి. కొంతమంది రెస్పాన్స్, దేర్ ఇస్ నో రెస్పాన్స్ ఫ్రమ్ సమ్ స్టూడెంట్స్. Okay, send your work to this number. Okay, bye to everyone.